Now in our previous classes, we have seen the method of spermatogenesis, oogenesis and the process of fertilization. Now after the formation of the zygote, what are the changes which are happening, we are going to discuss now. When you take the total duration of pregnancy, in case of human beings, it's normally 280 days or 40 weeks. Now this period is called as the gestational period, which is divided into three phases, one as a germinal period, two as the embryonic and third as the fetal period. The germinal period normally extends from 1 to 3 weeks, and this results in the formation of the zygote and for the process of the implantation. The second stage is the embryonic period, which is normally extending from 4 to 8 weeks, during which the differentiation of germ layers occur and appearance of the cylindrical embryo will be seen. The embryo is recognizable in human-like appearance, and this usually measures 2 to 3 centimeters from head to tail. The third is the fetal period, which is extending from third month till the termination, during which the rapid growth of the fetus will occur without any tissue differentiation. Now the first stage, which is again divided into 3 weeks, first week, second week and the third week. So there are changes which are occurring in each week. So today now we are going to describe the changes which are occurring in the first week of pregnancy. From the fertilization to the implantation, this is the thing which is happening during the first week. Now during this process, there is something called as cleavage takes place. Now what is cleavage? Cleavage is the repeated mitotic divisions of the zygote which result in rapid increase in the number of cells that are called as the blastomeres. This whole process occurs in the fallopian tube and particularly in the ampulla of the fallopian tube. Now the single cell zygote will divide into 2 cell stage post 30 hours after the fertilization, and next it will become 3 cell stage after 40 to 50 hours, 12 cell stage at 72 hours and 16 cell stage at 96 hours. As you can see in these pictures, this is the 2 cell stage, the 4 cell, the 16 cell which will be going into this. This is the stage of the morula. The 16 cell stage is described as a morula. The cells which are present in this morula are called as the blastomeres, and it has a mulberry-like appearance. And this is surrounded by a thick membrane which is called as the zona pellucida. Now this enters into the uterus nearly 3 days after the fertilization, and the factors which are helping for this movement are the ciliary beads and the contraction of the musculature of the uterine tube. Within the morula, it contains an inner cell mass which is called as the embryo proper and the outer cell mass which are forming the coverings of the embryo. As morula reaches the uterine cavity, fluid from the uterine cavity penetrates the zona pellucida and accumulates in between the inner cellular masses. Now a single cavity is formed, and that is called as the blastocele. The morula after the formation of the blastocele is called as the blastocyst. This takes place on the 4th or the 5th day after the fertilization. The blastocyst consists of the inner cell mass which includes 8 cells which in future will form the embryo proper, and that is called as the embryoblast, and the outer cells are 99 in number which are called as the trophoblast, which result in the formation of the placenta and the coverings of the embryo. Later the zona pellucida disappears, allowing the implantation which is done usually by the trypsin enzyme. The outer 99 cells which are called as the trophoblast are again divided into 2 types. One is the polar trophoblast which are normally 30 in number, and the mural trophoblast which are 69 in number. The polar trophoblast are the cells which are present near the embryoblast, and the remaining cells which are present at the periphery are called as the mural trophoblast. Now coming to the clinical application of this, the embryonic stem cells are derived from the inner cell mass, and these cells can give rise to any type of tissues, and they are called as the pluripotent cells. Now after the formation of the blastocyst, it goes into the process of implantation. Now what is implantation is nothing but the penetration of this blastocyst into the superficial compact layer of the endometrium of the uterus. The time when this implantation occurs is normally 6th day post fertilization, and it is usually completed by 11th day. The site of the implantation is normally the fundus of the uterus, normally in the middle line. The endometrium after the implantation is called as the decidua. The main goal for this implantation is to provide the nutrition of the embryoblast because the yolk material has been completely utilized during the cleavage and blastocyst formation. The trophoblast cells play a key role in this process. Now they are differentiated into two types of cells. One is a cytotrophoblast, which is the inner layer, and the outer syncytial trophoblast. This process occurs during the invasion into the uterine mucosa. Now coming to the stages of implantation, the first stage where the attachment of the blastocyst occurs to the endometrial epithelium. It follows the zona pellucida lysis. The blastocyst attaches to the endometrium with the embryonic pole. The second stage where invasion of the trophoblast cells in the endometrium occurs. This follows as a result of the contact of the trophoblast with the endometrium. The trophoblast cell starts differentiation in the entire blastocyst. It needs about 3 days, that is on the day 10th post fertilization, and this is the period where the implantation will occur in the endometrial stroma. The third stage is the time where the reparation of the implantation defect in the endometrium occurs. After the penetration of the blastocyst, the defect in the epithelium is filled with a closing plug, which is called as an operculum. This is nothing but a blood clot and a cellular debris which occurs by day 12. The site of the implanted conceptus appears as a minute elevation of the endometrial surface, and it is covered with the regenerated epithelium. Now in the fourth stage, there is a formation of the uteroplacental circulation which occurs around day 9. During the day 9, there will be isolated spaces called as lacunae or formed within the trophoblast. They become filled with the nutrition, secretions which are eroded from the endometrial glands and with the maternal blood from the ruptured maternal capillaries. This occurs between day 11 and day 13. The adjacent lacunae fuse to form a lacunar network in which the maternal vessels will be opening, through which the maternal blood will be flowing. Now there is a fifth stage where there are changes which are occurring within the decidua. The conceptus evokes reaction of the entire endometrium of the uterus. It consists of the endometrial stromal cells that enlarge and accumulate glycogen and lipid droplets, and there will be glandular alterations. The uterine glands increase their activity. These changes are being normally described as a decidual reaction. The decidual transformed endometrium is known as the decidua. Now, what is decidua? Decidua is nothing but the compact layer of the endometrium after the implantation of the blastocyst. There are 4 types. One is the decidua basalis, decidua paritalis, decidua capsularis, and decidua marginalis. Now, what is decidua basalis? This is nothing but the part of the endometrium between the blastocyst and the myometrium. So, this part is described as decidua basalis. Now, coming to the decidua paritalis, this is nothing but the endometrium which covers the wall of the uterus. The decidua capsularis is nothing but the endometrium between the blastocyst and the uterine cavity. And the last is the decidua marginalis. This is nothing but the endometrium surrounding the site of implantation. So, this is the decidua basalis, this is the decidua capsularis, decidua marginalis, and this is all the decidua paritalis. Now, coming to the implantation sites. The normal implantation is occurring at the fundus of the uterus. Besides that, there are some other sites where the implantation can occur, which results in the ectopic pregnancy. The normal site of implantation is normally the fundus of the uterus. Besides that, it can occur anywhere, which results in the tubal pregnancy or the ectopic pregnancy. Out of the total ectopic pregnancy, tubal ectopic pregnancy is 90%. and usually these tubal pregnancies results in rupture of the fallopian tube or the uterine tube and hemorrhage during the first 8 weeks, followed by the death of the embryo. Implantation in the ovary is called as the ectopic ovarian pregnancies. Ectopic pregnancies are connected with profuse hemorrhage. There may be sometimes in the uterine ectopic pregnancy where implantation will occur at the cornua of the uterus, which is the lateral angle, which results in abortion, or sometimes in the lower part of the uterus, which is called as normally placenta previa, or within the cervix. Besides that, there are some extrauterine ectopic pregnancy cases where the implantation which occurs outside within the peritoneal cavity, and that is called as the abdominal pregnancy. Till now we have seen the changes which are occurring in the first week. So first week is the time where the implantation will occur, and this implanted tissue will have an inner mass which is called as the embryoblast and the outer cells which are called as the trophoblast. The embryoblast during the second week will differentiate as an embryonic disc, which gives rise to the germ layers that form the various tissues and the organs of the embryo. And the outer trophoblastic cells will be forming the extraembryonic structures like the amniotic cavity, the amnion, the umbilical vesicle, the connecting stalk, and the chorionic sac. Now we are going to see the changes in the second week which result in the formation of all these structures. At the time of implantation, we have a group of cells inside which are called as the embryoblast, and the group of cells which are present at the periphery which are called as the trophoblast. Now we'll see the changes which are occurring in the embryoblast. The cells of the embryoblast are now differentiated into 2 layers, which occurs normally after 8th day after the fertilization. The inner cells are now differentiated into 2 types of layers, are the hypoblast and the epiblast. The hypoblast are the flat cells which will in future will form the primary endoderm. The epiblast cells are the upper layer of cells which are tall, columnar, and in future they will be forming as a primary ectoderm. The ectodermal cells now separate from the cytotrophoblast, which are the peripheral cells, and result in the formation of a cavity called as the amniotic cavity. The yolk sac is formed normally on the 9th day. Now coming to the boundaries of this yolk sac, the yolk sac is bounded by the hypoblast or the endoderm and the thin membrane which is called as the Hughes membrane. The Hughes membrane is formed by the flattened mesothelial cells at the embryonic pole. The primary extraembryonic mesoderm is forming during this time between the trophoblast externally and the amnion and the primary yolk sac internally. The extraembryonic coelom, which is formed outside, now splits into 2 parts as a somatopleuric and the splanchnopluric layer. The somatopleuric layer lines the cytotrophoblast and the amnion, and the splanchnopluric lines the yolk sac. The amnion is connected to the blastocyst by a cellular cord called as the connecting stalk or the body stalk, which in later forms rise to the umbilical cord. The chorion is nothing but the trophoblast with the somatopleuric layer of the extraembryonic mesoderm. The primary extraembryonic mesoderm is formed between the trophoblast externally and the amnion and the primary yolk sac internally. Now this extraembryonic coelom is now divided as somatopleuric and the splanchnopluric layer. The somatopleuric layer lines the cytotrophoblast and the amnion, and the splanchnopluric lines the yolk sac. The amnion is connected to the blastocyst by a cellular cord called as the connecting stalk or the body stalk. This in future will form as the umbilical cord. Now there is a formation of the chorion. The trophoblast with the somatopleuric layer of the extraembryonic mesoderm is now called as the chorion. There will be a part of the yolk sac which is getting separated and which is described as a secondary yolk sac. The secondary yolk sac is usually present at the embryonic pole, and this is formed on the 13th day after the fertilization. Now, within the endoderm, there will be changes will be occurring which result in the formation of the precaudal plate, and this end will form as the cephalic end of the embryo. Now, the changes which are occurring in the outer mass, that is nothing but the trophoblast. After 8 days after fertilization, the trophoblast differentiates into 2 layers. The inner layer is called as the cytotrophoblast and the outer layer is called as the syncytiotrophoblast. The inner cytotrophoblast are mitotically active cells, and we can see the cell boundaries. And the outer syncytiotrophoblast has a rapidly expanding multinucleated mass in which no cell boundaries are described. Within the syncytiotrophoblast, the lacunar spaces appear, and these lacunar spaces are connected to each other. Within the syncytiotrophoblast, lacunar spaces appear, and in between the lacunae, the syncytial cells present as trabeculae. The lacunae are more normally at the embryonic pole. The lacunae erode the spiral branches of the endometrial arteries and endometrial veins. Now, the lacunae are filled with the maternal blood. This results in the formation of the uteroplacental circulation. Now, there will be formation of the villus. Now, during the 2nd week, there is a formation of the primary stem villus. What is primary stem villus? The cytotrophoblastic cells which are present as an inner mass, they go in between the trabeculae and form an outer cytotrophoblastic shell. This is called as the primary villus. The lacunae are called as the intervillus spaces. In the early part of the 3rd week, the outer cytotrophoblastic shell is formed. The syncytiotrophoblast produces a glycoprotein hormone, that is HCG or human chorionic gonadotropin, which enters into the maternal blood via the isolated cavities, that is the lacunae, which are present in the syncytiotrophoblast. HCG maintains the hormonal activity of the corpus luteum in the ovary during the pregnancy. The corpus luteum, which is an endocrine glandular structure that secretes the estrogen and progesterone to maintain the pregnancy, the work of that is taken by the HCG, which is produced by the syncytiotrophoblast. Now, coming for the pregnancy test, which is a highly sensitive radioimmunoassay which is available for detecting the HCG, and they form the basis of the pregnancy test. Enough HCG is produced by the syncytiotrophoblast at the end of the 2nd week to give a positive pregnancy test, even though woman is probably unaware that she was pregnant. Now, to prevent implantation, there are various methods. A high dose of diethylstilbestrol given daily for 5 to 6 days may also accelerate passage of the cleaving zygote along the fallopian tube or the uterine tube. The large amount of estrogen produced now disturbs the normal balance between the estrogen and progesterone that is necessary for the preparation of the endometrium for implantation. IUDs or the intrauterine devices usually interfere with implantation by causing a local inflammatory reaction. Some IUDs contain progesterone which is slowly released and interferes with the development of the endometrium so that implantation doesn't usually occur. Other IUDs contain a wrap of copper wire, and this copper is directly toxic to the sperms and also causes uterine endothelial cells to produce substances that are also toxic to the sperms. Finally, during the second week, within the embryoblast, we have the formation of two germ layers. One is the epiblast, which is the future ectoderm, and the hypoblast, which is the future endoderm. And within the hypoblast, there will be formation of the procaudal plate, which signifies the cephalic end of the embryo. And coming to the outer part, that is the trophoblast, is now differentiated into cytotrophoblast and the syncytiotrophoblast. And there will be formation of the primary stem villi, which has the inner cytotrophoblast surrounded by the syncytiotrophoblast. And the lacunae, the spaces which are present in between the syncytiotrophoblast, are filled with the maternal blood and results in the uteroplacental circulation.